Good morning. My name is Dr. John Thomas. I'm an associate professor in the Department of Human Genetics at the University of Texas Rio Grande Valley. I'll be conducting the first of the short course sessions today, and we'll be starting with a basic overview of the immune system and the immune response in general. So our learning objectives for today are to identify some of the primary features of immunity, be able to distinguish between the two main phases of the immune system, the innate and the adaptive immune response, and we'll briefly touch on some mechanisms of pathogen clearance. So what is the immune system and immunity? Essentially, the immune system is a biological system for the identification and the elimination of pathogens within a larger organism. So we'll define some terms here that we'll use throughout the talk to kind of keep everything relevant. I've got these highlighted and listed down the bottom of the slide. These are pathogen, antigen, antigen presenting cell or APC, and presentation. A pathogen is a general term that I will use throughout the talk to refer to any agent that could cause a disease. These could be any kind of microbial agent, viruses, bacteria, but also could be fungal. These could be parasitic worms. These could be molds. Any agent that could elicit a disease response inside of an organism, we will refer to under a very broad term as a pathogen. An antigen is going to be used as a term to cover substances that elicit some kind of an immune response. These could be proteins, these could be glycoproteins, these could be carbohydrates, but these general terms, this general term antigen will be used to describe any substance that's going to elicit an immune response. An antigen presenting cell is a cell responsible for displaying or presenting antigen to the various cells of the immune system. And the term presentation within the context of immunity is used to describe how antigen is displayed or presented within the context of an antigen presenting cell. So these will be some basic terms that we'll cover when we discuss the immune system and immunity. What are some of the features of the immune system? It's one of the most ancient and widespread systems of all the biological systems that we encounter on Earth. Virtually every multicellular form of life on our planet has some form of immune system. The hallmarks of the immune system can vary between species. And for example, we see a bit of a different reorganization of the immune system in invertebrates than we do in vertebrate animals. But nearly every species on the planet that's a multicellular life form has some form of immune system to protect it against pathogens. Immunity is a great example of a distributed system that's been built from very complex components that are self-renewing. So one of the hallmarks of the immune system is it has the ability to replenish itself. We'll talk a little bit in a few minutes here where the immune system cells come from, but the immune system has the ability to continually renew itself in the face of adversity, in the face of being used and challenged over and over. What are some of the key hallmarks of immunity? One of the hallmarks of the immune system is that it's very adaptive. It's able to train the self, to train the immune system, the body, to respond to new threats. It's error tolerant. This means that if the immune system makes a mistake, 99.9% of the time, the mistake is not going to be a fatal mistake to the organism. So there's some flexibility built in the system. The immune system is able to engage in what's called self-protecting. It can protect itself from pathogens, from foreign invaders that might try to circumvent or eliminate parts of the immune system in order to be able to establish infection. And lastly, the immune system has memory. It has the ability to be able to remember a previous pathogen encounter and then respond quickly upon reintroduction. And we'll talk about some of the hallmarks of immunity and memory here as we get further on in the discussion. The immune system is essentially designed to protect against 4 different types of pathogens. From a very broad sense, you could look at these pathogens as being pathogens that are going to be extracellular. In other words, pathogens that have been able to get inside the organism, but they are not replicating inside of a cell. And pathogens that are intracellular. When we commonly think of intracellular pathogens, pathogens that will replicate inside of a cell, we generally think of viruses fulfilling this role, although there are some bacteria that also replicate intracellularly as well. But essentially, we've got 4 classes of pathogens the immune system is going to provide protection against. Viruses, which, as I said a few minutes ago, are going to be replicating in an intracellular environment. Bacteria that can replicate in an intracellular environment, as well as some types of protozoans and parasitic organisms. Extracellular bacteria, these would be parasitic worms, fungi, and then parasitic worms that are extracellular in general. So we've got 4 types of pathogens that the immune system has been essentially designed to provide protection against. So these cells of the immune system, where do they come from? Where are these components of the immune system that have this ability to be self-renewing? Where do they come from? Well, they're derived from cellular precursors. This is a very fancy way of saying that these cells of the immune system come from other cells that themselves come from the bone marrow. So we basically have a class of cell that matures and grows within the bone marrow and then can leave the bone marrow. And as it leaves the bone marrow, depending on where it ends up in the body, it can differentiate into different types of effector cells based upon the environmental conditions. And many of these functions are tied to a specific response. So if we look at the image up here, this is showing us essentially the red marrow from a bone graft perhaps. We have this cell that comes out of it that's called the hematopoietic stem cell, which then as it circulates to the body, and depending on what compartment within the body it ends up in, it can differentiate into different cell types, which can then go on and differentiate into other cell types. And all of these are tied to carrying out a specific function within the immune system. If we look at this from a cartoon perspective, at the top we see bone marrow, and this is going to be our progenitor cell that's going to come out of the bone marrow. We technically refer to this cell as a pluripotent hematopoietic stem cell. And this mother cell, if you will, that comes out of the bone marrow, then has the capacity to differentiate into all these different cell types that we see up here on the screen. So if we look on the left-hand side of the tree, we see that the bone marrow cell becomes a common lymphoid progenitor. It then can differentiate into B cells, T cells, and natural killer cells. And then as these naive cells make their way to different areas in the body, such as the lymph nodes, they will then be converted into effector cells that then have the ability to go out and carry out a specific function within the immune system. On the other side of the tree, we see cells, this pluripotent hematopoietic stem cell that remains in the bone marrow will then differentiate into different effector cells that are tied to other responses in the immune system. So we see, for example, the pluripotent hematopoietic stem cell becoming a myeloid progenitor cell. And these cell types then have the ability to differentiate into erythroblasts, platelets, as well as some of these smaller effector cells that normally circulate through the blood, such as neutrophils, eosinophils, and basophils, and monocytes, which will ultimately be used to either help drive an immune response against a specific pathogen. In the case of parasitic worms, we're maybe looking at eosinophils, for example. And we also have the ability for these cells to differentiate another time, in the case of the monocyte, to become an effective cell known as the macrophage, which now has the ability to move throughout the body, ingesting foreign objects or foreign substances that it comes across, and then being able to examine these foreign objects to see whether or not they are worthy of generating an immune response against, or if they are from the cell. So we have these cells from the immune system are all coming and derived from a mother cell, this pluripotent hematopoietic stem cell that initially leaves the bone marrow before it diffuses throughout the body and can differentiate into these various effector cells. But all of the cells in the immune system are derived from this single cell that comes out of the bone marrow. So just taking a look at some of the basic functions of the cells of the immune system, and I like to put this graph up here because this gives you the ability to kind of correlate what the actual image of the cell looks like compared to the cartoon that we use to represent what the cell looks like. So if we look at the top, we see macrophages. These are going to be the cells that are primarily involved in phagocytosis, cellular eating, if you will, and they will be taking up foreign antigen, foreign substances within the body, and then engaging in antigen presentation, which we'll talk about here in just a moment. Another example are eosinophils. These are cells that are able to kill parasites that have been coated with antibodies. We have dendritic cells, for example. These are referred to sometimes as the generals of the immune system. They help coordinate the innate and adaptive immune response, which we'll talk about here in a moment. Very important antigen presentation cell. Basophils, neutrophils, mast cells. So all of these cells are going to carry out a very specific function within the immune system. And again, I just kind of like to put this image up to show you that at least when we look at the cartoon, there is a bit of small connection, however remote, between how the cartoon image looks and how the cell actually looks under a microscope. So when we talk about the immune system in a very general perspective, one thing that we can do is we can kind of help to stratify and categorize the immune system and the immune response into basically two arms. We refer to these arms or these lines of defense as innate and adaptive immunity. So we're going to talk briefly about these two arms of the immune system and what their roles are in helping provide protection from a given pathogen. So from an immune perspective, the first line of defense is often going to be the innate immune response. And this is essentially a series of barriers that the pathogen has to overcome in order to be able to establish an infection. So if you can prevent the establishment of infection, if the pathogen is unable to interact with and defeat the innate immune response, then you're not going to have the establishment of infection. So this is really going to be the first arm of the immune system that the pathogen has to overcome in order to be able to establish infection. And when we look at the innate immune system, what we're essentially talking about is, by and large, not 100%, but by and large, we're talking about a series of barriers. Barriers that are going to be physical, they're going to be mechanical, and they're going to be chemical. And again, these are going to be the first barriers that pathogens will encounter prior to their attempt to be able to establish infection within a given host. And so if we look at the top of the graph, we've got skin, gut, lungs, and the eyes and nose and oral cavity as locations within the body. And then moving down the y-axis, we have the different types of actions within those areas, mechanical, chemical, or microbiological, that help aid the immune response in preventing pathogen infection and the establishment of infection. So we have this general defense system that's going to function in a very non-specific way in order to help prevent the establishment of infection. The innate immune system is going to use things such as the tightness of your skin. If you look at your skin, for example, your skin is normally an unbroken barrier of keratinized cells that are lightly covered with wax and oil that is by and large water resistant and resistant to penetration from most substances. So if the tegmental layer of your skin is intact, then it's going to be very difficult for a pathogen to gain access to the tissues that are underneath the skin. So having intact skin is going to be very important in helping prevent initial exposure to pathogen and helping prevent infection. If we look a little deeper and we look at areas such as the gut or the lungs, then not only do we have these mechanical barriers where the cells are very tightly joined and very closely intercalated together, but we also have things like the flow of air or fluid over areas within the gut or areas of the skin. So these things are going to help by providing a constant source of agitation, which makes it difficult for a pathogen to establish infection. If you think about the analogy of a flowing river, when water is still and the water doesn't move, this is when water tends to become brackish, it becomes dirty. We see the buildup of silt, of bacteria, of plankton, for example. But if you compare that to a river that is a flowing river, you don't find these areas where the water is still and built up because the water is constantly moving and it's keeping everything fresh and it carries dirt and debris and other things that can help maybe benefit the establishment of infection away from the body, if you will. So having the flow of air and fluid over the skin within the lumen of the gut helps prevent pathogens from being able to physically establish a foothold in order to be able to initiate infection. The same thing happens in our lungs and our oral cavity. We have cilia that are helping to move substances around. We have the production of tears. We have the production of surfactant. So we start to look at then what are some chemical advantages that the innate immune system has in order to be able to prevent infection. Well, we've got the production and the presence of defensins, of fatty acids within the skin that are going to help in a very non-specific way prevent pathogens from being able to establish infection. Within the gut, we not only have an enzymatic cocktail of enzymes that are designed to help break down proteins such as pepsin, but we also have a very low pH environment. I already mentioned the enzymes in tears and the surfactant is in the lungs. So we have a layer of chemical defenses that work with the mechanical defenses to help prevent the establishment of infection. And then if you look at from a microbiological perspective, the normal microbiota that we always have inside of us, the bacteria that live in our guts, maybe we've got some bacteria that have colonized symbiotically some of our pulmonary airways, some of the nares, and the presence of these bacteria there that are part of the normal flora for a human being helps prevent the establishment of infection. If the bacteria are already living in the gut, they're going to have created a unique environment that benefits their replication. And it's not in their best interest to have an environment that will allow other bacteria to come in and be able to set up shop and replicate within their ecological niche, if you will. So we have a series of physical barriers that are working along with the presence of some chemicals to provide a very general, non-specific line of defense against invading pathogens. And this is really what innate immunity is a non-specific line of defense against these pathogens. Now, in addition to having physical barriers and mechanical barriers in the production of some chemicals, we've got a cluster of receptors that are scattered throughout the body, particularly on the surface of cells, as well as within some of the vesicles inside of cells that can help drive and amplify the immune response when they perhaps do come encounter with a pathogen. And we refer to this general receptor series as toll-like receptors. This is basically a cluster of receptors that are designed to recognize unique components of foreign pathogens. If you look at the list, for example, we have some TLRs, toll-like receptors, that are bound on the surface of cells, such as TLR4, which is able to recognize lipopolysaccharide, LPS from gram-negative bacteria, one of the primary components of the cell wall from a gram-negative bacteria. We have some TLRs, such as TLR2, that recognize other peptides from bacteria. So you have these toll-like receptors that are on the surface of cells that are designed to kind of provide an early warning system for the detection of components of bacteria. As well, we have some TLRs that are located on the inner leaflet of the endosome that are designed to detect against agents or materials that might be coming from intracellular foreign invaders, such as viruses. So TLR3, for example, is a toll-like receptor that specifically responds to the presence of double-stranded RNA, which is a very common molecule that you will encounter when the cell is infected with an RNA virus, such as polio or dengue virus, for example. We also have TLRs that can detect virus from RNA from single-stranded RNA viruses. We have TLRs that can detect unmethylated DNA from bacteria and viruses. So we have another array of receptors that, in a somewhat non-specific way, they're specific in the sense that they're going to respond to a certain type of viral RNA or certain type of bacterial DNA, but they're general in the sense that they're not going to distinguish between generating an immune response against Yersinia pestis versus E. coli. They're both gram-negative bacteria, and from a TLR's perspective, they're going to respond to the LPS using TLR4. So there's not quite the specificity yet that we see with the other parts of the immune system, but we do see within the toll-like receptor family an attempt by the immune system to kind of further tighten the immune response from being a very broad response of such as the skin, where we're stopping all pathogens from coming in, to now having a series of receptors that can perhaps not identify and respond to a specific pathogen, but can respond to a class of pathogens, such as viruses, RNA viruses, or intracellular bacteria, for example. So the toll-like receptors are a very important phase of the immune system, particularly for helping the innate immune system respond in a very rapid but non-specific way to a pathogen. So when we look at the innate immune system from a 10,000-foot view, assuming that the innate immune system is functional and intact, and we're talking about a healthy organism, very few pathogens are going to be able to evade the innate immune response. But assuming that they can, then we have another arm of the immune system that can take over and step in to help drive a response to the pathogen that will be beneficial to the host and hopefully lead to clearance of the pathogen. And we refer to this other arm of the immune system as adaptive immunity. So if we look at the flow chart that we've got on the left-hand side of the screen, we see we have the pathogen coming in at the top, and then the pathogen encounters some kind of an anatomical barrier. Let's say this is the skin, for example. You've got some bacteria that's landed on the surface of your skin on your arm. Assuming that your skin is intact, there's no open sores or cuts, which would give the bacteria access to the fluids and the cells underneath the skin, you're going to have infection prevented. If, however, that anatomical barrier containment fails, then you'll get the establishment of infection. So now you've got infection established under the skin, but you still have a system of preformed, non-specific, and broadly specific effectors that can come in and respond to the pathogen. These would be things like the toll-like receptors within the cells that are encountering pathogen. And hopefully this would lead to the removal of the infectious agent. If, however, this still fails and you now have the beginning of the establishment of infection, then you're going to lead to the recruitment of effector cells, and now you're going to get what's called the early induced innate response. And this is going to be the immune response that most people are going to generate within the first 3 to 4 days of encountering a pathogen. And this is going to hopefully give your body time to respond to the organism by keeping the numbers of the organism that's replicating in the case of a virus or a bacteria, or really any pathogen that's gained access to the host. It's going to begin to replicate. And at this point, what your innate immune system is trying to do is to keep the numbers that are being generated low enough to when your adaptive immune response kicks in 4 to 5 days later, that it has the ability to be able to start to bring those numbers down. So in many respects, when you look at a pathogen invading a host and trying to establish infection, it becomes a numbers game. Can the pathogen replicate to a level that's sufficient to be able to overwhelm the immune system and establish infection? And from the immune system's perspective, its job is to try to get those pathogen numbers down from potentially billions of pathogens, if we're talking about viruses or bacteria, down to millions, down to 1 million, down to hundreds of thousands, and then thousands, and eventually then they'll eliminate the pathogen. So we've talked about the innate immune response, but for whatever reason, the pathogens been able to circumvent innate immunity and establish infection. And so now we're going to have to rely on the other arm of the immune system called the adaptive immune response to now step in and take control. So when we talk about adaptive immunity, just as the name implies, with innate immunity, we're now talking about an adaptive immune response. This is an immune response that's going to be specific to the pathogen coming in. So you are infected with dengue virus, not just a virus, but with dengue virus. You're now going to have an adaptive immune response. It's going to generate an immune response that's going to be specific to dengue virus, not just all viruses in general, but to dengue virus. How does the adaptive immune system do this? We'll discuss this here in just a minute. When we look at the adaptive immune response, we're really talking about kind of two classes or two components of the adaptive immune response. Again, from a very 10,000-foot view, that are really going to help drive adaptive immunity. And these are B cells and T cells. So we're talking about a cell that's going to come in, the B cell, and that's going to produce antibodies that can help to neutralize foreign pathogens. And then we have what's called a cell-mediated response driven by T cells, which will come in and specifically target cells that are infected with pathogens. And again, as I said a few minutes ago, you really are looking at the innate and the adaptive immune responses coming together within a single host and working together to eliminate pathogens while also preventing any deleterious effects on the host from the immune system response. We'll talk in a few minutes about what happens when the immune system doesn't function properly, but we are again talking about a system within our body that's designed to kill not only invading pathogens, but infected cells as well. So you can imagine a scenario where the immune system loses the ability to differentiate between an infected cell and an uninfected cell, and this could have negative outcome for the host. And we'll talk about what happens when the immune system loses tolerance, when it loses the ability to ignore self and instead focus on the pathogen as well. So when we're talking about the adaptive immunity, we're talking again about B cells and T cells. Another way that this is commonly referred to is humoral immunity and cell-mediated immunity. Again, humoral immunity refers to the production of antibodies through B cells, and cell-mediated immunity really refers to the actions of T cells directed against infected cells. So let's talk a bit more about the adaptive immune response. So as I mentioned a few minutes ago, the immune response in totality is going to be a phasic response. Sometimes this is referred to as a biphasic response because you're really getting two phases of immunity that are working together. The innate immune response, which is going to be our rapid, non-specific response to infection, and then the adaptive immune response, which is going to be a not quite as rapid, but very specific and very focused response to infection. So if we look at the face of the immune response and we talk about the innate immune response, we're really talking about a response that's going to take minutes to respond, but it's going to take days in its ability to be able to clear the pathogen. Again, the innate immune response is a very rapid, robust response, but it's not specific. So the response could take several days to be able to ramp up to clear the pathogen if it's just relying on the innate immune responses by itself. When we talk about the adaptive immune response, then we're talking about a response that takes anywhere from hours to a few days to begin to really ramp up to activate. And this adaptive immune response can last anywhere from a few days to several weeks. And within the adaptive immune response, this is where we're going to get the specificity within immunity that we often think about when we talk about the immune system in general. So this is going to be cells of the immune system that are specifically focusing on eliminating that dengue virus infection that you have. So they're going to go after cells that are infected with dengue virus, dengue virions that are floating throughout the body, any phagocytic cells, any cells that have taken up dengue virus at all these are going to be eliminated through the adaptive immune response. And then the last phase of the immune response, once we've cleared the pathogen, is going to be the generation of what's referred to as memory or immunological memory. So this is going to be, just as the name implies, a memory of the event. The adaptive immune response is going to remember that it encountered dengue virus. Remember that it had to clear this specific type of dengue virus. Dengue virus comes in different flavors and different serotypes. So not only is the immune system going to be able to say, hey, we just had to respond to a dengue virus infection, but ideally it will say, hey, we just had to respond to this serotype 1 dengue virus infection. And so now we're going to remember what the response was. So when we have another infection with dengue virus occur in the future, if it's the same serotype, we can have a very rapid response, which instead of taking weeks to build up, can start to work in days. And this is a response and a level of protection that can be lifelong. This immunological memory is the theory behind why we use vaccines to drive long life protection against certain pathogens. It's based around this concept of immunological memory, that you're going to have B cells and T cells, the cells of the adaptive immune response that produce antibody and that go after infected cells. You're going to have a population of these memory cells that are going to remember the response against dengue virus. And then if you encounter dengue virus in the future again, the response can be much quicker and much more robust than it was the first time. So these are the three phases of the immune response. And again, you're talking about a response that can take minutes to occur without any specificity, which is the innate immune response, which is going to bleed into the adaptive immune response, which can take days to weeks to occur, but which can be a very robust response. And the end goal of the adaptive immune response will hopefully be the generation of memory, which will help the host respond in the future should they encounter the same pathogen again. When we talk about some of the cells and some of the effectors, if you will, within the immune system, there's many, many cells that play very important roles within the immune system. But when we're talking about the adaptive immune response, we're really going to be looking at a few very specific cell types, and these are primarily going to be T cells. If we look within the effector modules for immunity, we are talking about we need cells that can help drive intracellular immune responses, the elimination of pathogens that are intracellular pathogens, as well as to activate other cells of the immune system to help magnify the response. We're talking about cells that need to be able to affect some kind of extracellular immune response. So these are going to be pathogens that have perhaps gained access to the host, but they're not intracellular yet. They're floating around in the interstitial space in the extracellular environment. So we need a cell that can go out there from the immune system and interact with those pathogens. And we also need cells that carry some general level of cytotoxicity, which is cells of the immune system that are able to go out there and interact with host cells that might be infected with virus or bacteria, or maybe they're just host cells that are extremely stressed. They are tired metabolically, and they just need to be turned over and eliminated. So we have these different cell types within the immune system that carry out these different effector roles of cytotoxicity, protection at mucosal surfaces, and then protection from both extracellular and intracellular pathogens. So how does this happen? So we spoke in the beginning of the talk about a term called presentation. And this is one of the hallmark components of the immune system, is this concept of antigen presentation. So we have a foreign invader that gains access to the host. This foreign invader, because it's foreign and is not of the host, it's not of the self, is going to carry with it proteins, carbohydrates, and nucleic acids, fats, a whole host of biological molecules that are not going to be of the self. They're going to be different. And so because they are different, by their definition, these are referred to as antigens. And so many of these foreign invaders, as they gain access to the host, are going to be producing a range of proteins, carbohydrates, and other molecules that are going to stimulate the immune response. Well, how do they do this? Well, they do this because located within these proteins or these carbohydrates that the foreign invader is producing are going to be specific regions, specific arrangements, physical arrangements that we refer to as epitopes, which will stimulate an immune response. So we have to have then a system within the body that can take these foreign substances, interact with them, grab them, for example, and then examine them, hold them up and look at them and say, is this really a foreign pathogen? Is this a foreign peptide? Or is this just a piece of me that I've never seen before? So this concept of grabbing the antigen and then presenting it for inspection is a very critical hallmark of the immune response, particularly the adaptive immune response. And so we have a host of cells within the body that carry out this phenomenon called presentation. And this presentation aspect is done through a molecule that's called MHC, or major histocompatibility complex. So if we look at the cartoon here to basically break this down, we have our dendritic cell indicated by the smiley green globby guy. He's going to phagocytose or eat a bacteria represented by the angry green circle. Once the antigen presenting cell, the dendritic cell, has internalized this bacteria, it's going to digest it, and then parts of the bacteria, parts of the dinner that the dendritic cell just ate, are going to be digested and then complexed with MHC and then presented on the surface of the dendritic cell. Once the dendritic cell has put this antigen piece of the bacteria on its cell surface, it can now present it. It can now show it to other cells of the immune system. And in the cartoon, we have a helper T cell that comes and interacts with this presented antigen. And this interaction then stimulates or activates the T cell. And now in cartoon 4, we see that our purple helper T cell is now activated. He's super happy. And now he's activated against the antigen that he just interacted with on the surface of the dendritic cell. And now through presentation, this T cell is now stimulated. he's ready to go out there in his active form and fight the infection. So antigen presentation is really going to help drive the specificity of the immune response against a given pathogen by taking a piece of that pathogen, complexing it with MHC, and then showing it to other cells of the immune system so they can then drive a concomitant immune response against that pathogen complex with MHC. So major histocompatibility complex, or MHC, is a very important molecule within the immune system, particularly within the context of adaptive immunity. Many times the epitopes, the sequence of peptides or carbohydrates in some examples that are needed to stimulate immune cells are hidden. They're within a deep fold in the protein. And so this means that the protein, if we're talking about a protein epitope, antigens located within a protein, that means that protein is going to have to be broken down into its individual peptide pieces. And then once it's broken down into its individual peptide fragments, those pieces of the epitopes, those epitope peptides, must be complexed with MHC and then sent to the surface of the cell for presentation. So this is a very tightly regulated process. The peptide, the foreign peptide, must be broken down. It must be complexed with MHC, and then it must be shown to another cell to be able to drive an immune response. If a pathogen is able to interfere with any step in this process, then it will block antigen presentation, and it will therefore block the immune system's ability to recognize the epitope and drive a response against the pathogen. And we'll talk about this in a later talk when we talk about mechanisms of immunoevasion and how pathogens escape the immune response. But in a normal, healthy environment, the epitopes are going to be complex with MHC. And just as is shown in the cartoon, MHC is now going to be presenting the epitope peptide from the pathogen to another cell of the immune system. In the cartoon, it's a T cell, and we have this recognition event happening between the T cell receptor, or the TCR. The receptor on the surface of the T cell recognizes the MHC molecule presenting the foreign epitope, and now the T cell can look at that and say, wow, this is not me. We need to do something about this. All of this is driven through MHC. We have two classes of MHC within the body, MHC class 1 and MHC class 2. MHC class 1 is found on virtually every nucleated cell type within the body. So for the purposes of our discussion here, are there some examples that push against this? Are there some I'm lost for the word. Are there going to be some cell types that won't have MHC? Yes. So this is not a completely blanket statement, but for the purposes of our discussion, all nucleated cells contain MHC class 1. Only professional antigen presenting cells, these are cells of the immune system, such as dendritic cells or macrophages, whose job it is to present antigen. Only cells of the immune system, certain cells of the immune system, carry MHC class 2. So every cell in your body carries MHC class 1, which gives every cell in your body the ability to show what's happening inside of it through MHC class 1. Only cells of the immune system that are professional antigen presenting cells are going to have MHC class 2. So where does this molecule come from? Where does MHC class 1 and class 2 come from? Well, these are molecules that are arrayed inside of the cell. So if we look at the cartoon here, we have a virus that's going to infect the cell. So viruses are intracellular pathogens. You could do the same and flip it for an intracellular bacteria, but for the purpose of our discussion here, we'll use the virus as an example. We have the virus infecting intracellularly, causing an intracellular infection. As a virus is an intracellular pathogen, the virus gains access to the inside of the cell, and it's going to start to synthesize viral proteins. As these viral proteins are being synthesized, we also have MHC class 1 molecules that are being produced within the cell. And as these MHC class 1 molecules are produced, they're going to be complexed with different pieces of protein that are inside of the cell. Well, because at that time, the cell is infected with a virus, some of those MHC molecules are going to be complexed with viral protein because we have viral protein present inside the cell because the cell is virally infected. So some of those MHC molecules, when they get pushed out to the surface of the cell, are going to have viral protein complex with MHC. So when we have viral proteins complex with MHC, and this makes it out to the cell surface, this is now going to lead to a signaling event where we're now going to have MHC complexed on the surface of the cell available for antigen presentation. And if we go back and look at our last cartoon here, when we have MHC complexed with our peptide and it's presented to a T cell, the T cell is going to look at that peptide and say, what is this protein? Is this from me? Is this a protein from inside the cell? Or is this a protein from a pathogen that's replicating inside the cell? And in our example, we have a cell that's been virally infected. And so when that MHC molecule, MHC class 1, because this is a standard cell, gets pushed out to the cell surface, it's going to have viral peptide mixed in with the MHC1 molecule. So when a T cell interacts with that MHC class 1 molecule, it's going to recognize that this cell is infected with a virus. This cell is sick. And so there's a specific response and treatment, if you will, that a sick cell gets that an uninfected or healthy cell does not get. But the way that the immune system can make this determination is by looking at what is complexed with MHC on the surface of the cell. So in the example that we have in the cartoon here, and this is mirrored at the bottom by the electron micrographs, we have our virally infected cell. It's been infected with virus. Now the virus is beginning to produce viral proteins. Inside the cell, we now have an infected cell. A T cell comes along. It interacts with this infected cell via MHC class 1. It sees that, hey, some of the MHC on the surface of this cell is mixed with viral protein. That means this cell is infected with virus. And this T cell, in this example here, this is going to be a cytotoxic T cell, an effector T cell that basically kills the infected cell. So through presentation of peptide with MHC, this is going to help the immune system decide whether or not it needs to kill an infected cell or if the cell is even infected at all. If MHC is presented to the cytotoxic T cell and it's self-MHC, then the T cell will leave that cell alone because it's going to say, you're not infected because you're showing me what's inside of you, and what's inside of you is self-protein. Again, if we have a virally infected cell, its MHC is going to complex with viral protein, and so that MHC, when it's displayed to T cells, is going to show what the intracellular environment is like within that cell. And if it's viral protein, then the T cell says, oh, you're infected, you have to be eliminated. So MHC recognition is how T cells will determine, how cytotoxic T cells will determine whether or not a cell is infected and needs to be killed, or whether it's uninfected and it can be ignored. MHC is critically important in driving these responses. So we can also look at MHC responses on the surface of antigen-presenting cells. So we have macrophages, for example. We have a cell of the immune system that basically goes through the body and scavenges and takes up and eats everything that it sees, and then it digests what it's eaten and then presents the digested fragments with MHC class 2 and puts it on the cell surface. And now T cells can come along and look at this macrophage and say, what did you eat for dinner? Did you eat something that's good or something that's bad? And again, from the immune system's perspective, good is going to be self-proteins, proteins from you, from the host. Bad are going to be foreign proteins. And since the macrophage is essentially the all-you-can-eat buffet guy that's going to run through the body and eat everything it sees, looking at T cells, looking at MHC class 2 on the surface of macrophages is a great way for the immune system to kind of get a quick snapshot of what's happening in the body at that time. So You've got MHC class 2 on the surface of antigen-presenting cells like macrophages that are going to help drive an adaptive immune response. And you also have MHC class 1 on the surface of every cell in the body that's going to let the immune system know what's happening inside of cells scattered throughout the body. So you've got a very, very nice level of coverage for both intracellular and extracellular in terms of what the adaptive immune response can generate a response against using MHC class 1 and class 2 molecules. So again, we have our infected macrophage. It's going to complex with maybe bacteria, maybe it complexed with maybe it phagocytosed some bacteria that were floating around. It's going to chop up the bacteria through digestion, complex the bacteria peptides with MHC. This gets presented to a T cell, and this example is a T helper cell, TH1, T helper is TH, and basically this T cell is now going to start to produce chemicals and generate a response to help this macrophage clear the infection and eliminate the pathogen. So the last cell that I really want to talk about in any great detail is the dendritic cell. And when we look at dendritic cells, they're commonly referred to as the general of the immune system. And why do we say this? Well, dendritic cells really are the link. They are the bridge, the gap filler between the innate and the adaptive immune response. If you go back and you look at the slide towards the beginning of the talk, we talked about lineages and where these cells come from. When we have cells, the pluripotent hematopoietic stem cell that comes out of the bone marrow can follow one of two main differentiation lineages, the myeloid or the lymphoid. The lymphoid are going to become the B cells and the T cells, and the myeloid are going to become the neutrophils, the eosinophils, the monocytes, the erythrocytes, the platelets, the red blood cells. Within those two phases of innate of the immune response, the innate, the non-specific rapid response, and the somewhat slower but very specific adaptive immune response sits the dendritic cell. And it has the ability to kind of walk in both worlds. It can help coordinate innate immune responses, and it can also help coordinate adaptive immune responses. The dendritic cell is an antigen-presenting cell, so it has the ability to phagocytose antigen, to break it down, and then present it. And depending upon the context that it presents the antigen, it can stimulate both innate and adaptive immune responses and actually work as a bridge and link the two responses together. So you don't have your innate immune response moving in one direction and the adaptive immune response moving in a separate direction. That coordination happens because of dendritic cells. Animal studies that have been done on mice that are have had different, that are transgenic, that have had different components of their immune system genetically removed from their body. When they've done this with things like dendritic cells, those mice don't live very long. They live for maybe a few hours. So the dendritic cell is a critical component of the immune response, and it really does function as this general, if you will, this link between the innate and the adaptive immune response. So dendritic cells particularly important within the immune response, just as important as B cells and T cells. So dendritic cells, as I mentioned a few minutes ago, have this ability to basically float around. They're hanging out in tissues all over the body, and the moment that they encounter an antigen, they will uptake the antigen through phagocytosis. They will break it down. While they're doing this, they will travel back to the lymph node. Excuse me, from wherever in the body they are, they will travel back to the lymph node once they phagocytose the antigen, and then they can start to interact with T cells through antigen presentation, present the antigen to the T cells. This then leads to the activation of the T cell, leads to some changes in the dendritic cell, and you see the induction of the immune response. So again, you really need to have dendritic cells able to be out there and kind of helping to direct the response and providing this connection between the innate and the adaptive response. So that way, from the host perspective, we're moving our immune system and our immune response in the most efficient way by targeting it to a single pathogen at that time. So let's suppose that you are a brand new unseen pathogen. You have never infected this person before, and somehow you've been able to evade the innate immune response. Perhaps there was a cut in the skin and you were able to evade the innate immune response. Should you be worried about adaptive immunity? If you are brand new and you've never invaded this organism before, how can any receptors bind to you if you're introduced into a new place? You should have free reign, right? It should be impossible for the immune system to respond against a pathogen it's never seen before. Well, of course, we know that this is not true based on the discussions that we've had. We have the innate immune response, which is going to provide some barrier. And even if you're able to avoid the innate immune response, there's still going to be things like dendritic cells that you're going to have to get over. But how does our immune system in general provide the ability to respond to a pathogen that you've never seen before? Let's say you're part of that group that goes with Elon Musk to Mars, and you're the first human beings to ever be on Mars. And perhaps there is a bacteria that we've never discovered before that's floating around in the dust on Mars. And you encounter that bacteria from an immune perspective for the first time. You've never seen that bacteria before. It's a extraterrestrial bacteria. It's ET bacteria. It's alien. How could you expect your immune system to be able to develop a response against something it's never encountered before? Well, the immune system has thought of this as well. And part of this is through the ability of the receptors of the immune system, particularly on B cells and T cells, to be able to generate a wide combination of receptors that can interact with different types of antigen. So if we look at this from the perspective of the T cell, we are talking about recombinations, and we typically refer to these recombinations as VDJ recombinations. If you look at the panel on the side, you'll see that we have V gene segments, D, which stands for diversity, and J, which stands for joining. And so we have these different gene segments that can come together in various combinations, and these different combinations bring a lot of different variations in the T cell receptor, or the TCR. The T cell receptor is actually going to be the part of the T cell that physically interacts with antigen when it's complexed with the MHC molecule, MHC1 or MHC2. So you're going to have this protein-protein interaction that's going to occur between the T cell and the cell that's presenting the antigen. And so in order for the T cell to be able to recognize that, it needs to have the ability to change its receptor makeup, to be able to go through some different combinations, because perhaps the receptor that the T cell has at that time doesn't physically merge with and interact with the MHC molecule. So T cell receptors have this ability to go through these different recombinations that we refer to as VDJ recombinations, and hopefully generate a high amount of diversity. Now, we've made some guesstimates as to the diversity within the T cell receptor, and it looks like, at least as of our last count, the different T cell receptor rearrangements that we can have are somewhere around 10 to the power of 16. So 10 to the 16th is the diversity repertoire that we have within the T cell. So nearly limitless diversity. So what this means is that your T cell receptor has the ability to generate virtually endless combinations. And these endless amounts of combinations give the T cell receptor virtually a limitless ability to be able to generate a receptor that can recognize that MHC. So if you're on Mars with Elon and you've got that Martian bacteria complex with MHC, maybe it forms a unique shape that your T cell receptor can't recognize at first, but the T cell receptor can keep going through these recombination events. And this is happening at the genetic level. And this is something we'll talk about in some future talks. We'll focus specifically on this mechanism. But this ability to go through these recombination events gives the T cell receptor a virtually endless amount of diversity. And this therefore then gives T cell receptors the ability to interact and recognize any peptide complex with MHC. From the host perspective, that's great in that we have all this diversity. The bad part comes from having to go through all those variations to get to the right one. If you imagine having a key ring with 10 to the 16 keys on it, and you're trying to find the key that best fits one lock in a door, you could be there quite a while going through this. This is one of the downsides of the adaptive immune response, is that it takes time for this response to get going. And this is why we need the innate immune response to kind of give us that time to potentially go through some of these recombination events to find the right key on the keychain to be able to unlock the door. But the receptor diversity within the T cells gives us a limitless ability to be able to interact with different types of antigen. So not only do T cells need to have the ability to recognize a vast array of different antigens, but we need to have some basic checks and balances on T cells as they are moving throughout the body. I talked at the beginning of the talk that T cells are going to basically interact with one of two types of MHC that are produced in the body. MHC class 1, which is present on virtually every nucleated cell in the body. There are some exceptions, but for the purpose of our talk, MHC class 1 is on every cell of the body. MHC class 2 is only going to be found on professional antigen presenting cells. So we have T cells that are going to be going around, and you could kind of think of the T cell in this role as kind of the night watch at the university. So everyone's gone home for the day. You're sitting in your office late at night, but all the other doors are locked. All the other lights are out. And here at UTRGV, we have the guard come through every hour, and he'll come through and walk through the building. And if he sees your light on, he'll probably come on. He or she will come over and probably knock on the door and say, "Hey, are you in there? Is everything okay?" You say, "Yes, I am." But you have the police coming through and doing their security check, making sure that the doors are closed, lights are turned off, nothing looks out of whack, and they move on to the next building. T cells are doing the same thing. They are coming through and they're checking every cell in the body. Only their door knock is MHC. The T cell comes along and interacts with MHC class 1 to see, hey, is this MHC class 1 foreign peptide, or is it mixed with self peptide? If it's mixed with foreign peptide, then this cell must be sick. We got to do something about it. But most of the time, the T cell is going to interact with self peptide complex with MHC. And this is basically the security guard doing the knock at the door. The T cell knocks, it says, "Oh, it's MHC with self peptide. Everything's fine. Let's move on to the next cell." But we need that interaction, that check, to go very smoothly. So when we have the T cell doing its safety check on the cell, if you will, its security check, the T cell needs to be able to do two things. It needs to be able to knock on the door, and it needs to be able to leave when it gets an affirmative response from the door knock. So the guard knocks on the door. We need the guard to be able to knock on the door. And we also need the guard to be able to hear us when we say, "It's okay, it's me working, everything's fine. You can go on to the next door." The way that T cells do this is through a process, through a phenomenon that we refer to as positive and negative selection. Positive selection refers to T cells that have T cell receptors, TCRs, that are able to recognize self MHC. So when a new T cell is made, a brand new baby T cell, if you will, it's going to check two things with its T cell receptor. Can it recognize self peptide? Is it able to say, "This is peptide from me, this is not peptide from me"? So positive selection is going to select from the baby T cell population T cells that can recognize self MHC. So this is the first check. Can this brand new T cell recognize self MHC? This is positive selection. The next step in the selection process is referred to as negative selection. So the T cell, the baby T cell, if you will, was able to recognize self-peptide. That's positive selection. How strongly does it interact with or react to that self-peptide? There's a golden spot where we're looking for. There's a sweet spot where we're looking for. We want the T cell to be able to recognize and differentiate between self-peptide and foreign peptide. That ability to differentiate between those two things is referred to as positive selection. The next part of the process is we need the T cell to be able to recognize self-peptide, but not stick around, not bind tightly to self-peptide. T cells that recognize self-peptide, but that bind very tightly to self-peptide are usually going to be eliminated from the body. And the way the reaction that the T cell has to the self-peptide is referred to as negative selection. So positive selection, we're selecting for T cells that have the ability to recognize self-MHC. Negative selection, we are removing T cells that bind and interact too strongly, too robustly to self-MHC. So we want to have a population, ideally, of T cells in our body that can differentiate between self and non-self. And we also want a population of T cells that when it does see self-peptide, not only does it have the ability to say, that's me, that's not me, but we need a T cell that has the ability when it sees self-peptide to say, that's not a problem, I'm going to walk away. If you have a population of T cells in your body that can differentiate between self and non-self antigen, but that react very strongly anytime they see self antigen, then you're going to have a population of T cells that are going to generate immune responses against the self, against the host, against the body. And this is what we think is the hallmark of many autoimmune diseases that we see, such as lupus and MS and other diseases of the immune system, is that we have T cells that lose the ability to regulate how tightly they bind to self-MHC. So the ability of T cells to recognize self-MHC and to not too tightly bind to self-MHC are generally referred to as positive and negative selection. And this is very, very important. It's extremely important that we have a population of T cells in the body that are self-tolerant, that can recognize the self and that don't flip out when it recognizes the self. We need these T cells to be self-tolerant. So positive negative selection is very, very important for T cells and the T cell response. So if we were to summarize the last two slides, we could basically say the immune system has a repertoire, it has a class of T cells that are capable of binding and destroying any cells that exhibit or produce any foreign protein that may be as a consequence of infection. So we've got this system in the body, these population of cells that are running around as the night watch. They're knocking on every cell in the body, they knock on every door and say, hey, is everything okay in there? If things are okay, it moves on to the next cell. If things aren't okay, it calls in backup and it eliminates that infected cell from the body. It could not only be an infected cell, it could be a cell that's diseased, that's worn out, that's tired, that's metabolically stressed. But for the purposes of the immune system discussion we're having today, we're talking about T cells being able to distinguish between healthy, uninfected cells and cells that are sick and infected, and then generate a response against the infected cell, not the healthy cell, but the infected cell, and then eliminate that cell from the body. So when we're talking about intracellular pathogens, most infected cells are going to be cleared by the binding and actions of the T cell. The first binding is going to lead to a stimulation of the T cell, is going to lead to T cell activation. That T cell is going to become active, is going to divide, it's going to generate and replicate and produce many copies of itself, and eventually recruit other cells of the immune system to that area with the goal of basically clearing the infection. So you have an intracellular pathogen, the infection's been detected by T cells have responded, they've mounted a response, they've produced chemicals that can recruit other cells of the immune system to the area. Everybody's come together in a big kumbaya moment, and the immune system has been able to eliminate the pathogen. So we've got our pathogen, Mr. Green cartoon. He says, okay, so I'm dead. You've been able to clear the pathogen. But what happens if a T cell binds to a non-infected cell? Will it also be destroyed? And the answer is, under normal conditions, no. And this is due to a phenomenon that we refer to as co-stimulation. So when our T cell sees a cell, it interacts with MHC class 1 or it interacts with MHC class 2, and it senses, hey, there's trouble here, then in order for the T cell to become active, it needs to receive two signals. It receives the danger signal, if you will, which is the antigen being presented with MHC. So we have our MHC in the cartoon, we have MHC class 2 on the surface of a professional antigen presenting cell. Let's say this is the macrophage. The macrophage has gobbled up a foreign invader. It's now presenting pieces of the foreign invader with MHC class 2 on the cell surface of the macrophage. A T cell comes along, interacts with that MHC class 2 and says, hey, this looks dangerous. We need to do something about it. I better activate. But before that T cell can activate, it needs to receive a second signal called a co-stimulatory signal that is kind of there to be able to confirm to the T cell, hey, this is a problem. You need to activate and start to mobilize a response. If the T cell sees the antigen, but it does not receive the co-stimulation signal, then that T cell becomes what we refer to as anergic, and it will not activate and clear the pathogen. So in order for T cells to become activated, it needs really two signals. It needs the signal and the stimulation from MHC interactions, and it also needs this co-stimulation signal. This is another mechanism by which the body and the immune system prevents T cells from just running crazy throughout the body and killing every cell that it sees. The T cells cannot be activated without the danger signal. But if it doesn't receive the danger signal and a second co-stimulation signal, then the T cell becomes tolerized to that antigen, and the effect from the immune system perspective is going to be no response. So there's an additional layer of protection that's built in to the immune system, to the adaptive immune system, particularly with T cells, that says, hey, you've got to get two signals in order to be able to become active and start to go out there and really start to ramp up the immune response. So we've kind of discussed about intracellular pathogens and the role of T cells in clearing intracellular pathogens. What if the pathogen is extracellular? What if it's a bacteria? What if it's a parasitic worm? So it's a pathogen that's been able to get through the innate immune response, and maybe it's floating around in the plasma or in the bloodstream, but it's not replicating inside of a cell. So if it's extracellularly replicating, it's in the extracellular space. How do we clear that pathogen? How do we generate a response against that pathogen if it's not inside of a cell? Remember, we just talked about the classic dogma of antigen presentation. The antigen has to be present within the cell to get complex with MHC so it can be pushed out to the surface to drive MHC signaling events. Well, if you're an extracellular pathogen, you're not going to be inside of a cell getting broken down and digested. So how does the immune system know that you're there? What does it do to respond against pathogens that are floating around in the bloodstream that are outside of the cell? And the immune system's response to this is the production of a molecule that's referred to as an antibody. So antibodies carry a basic structure. They're composed of two regions, the FAB, which is the part of the antibody. We can think of antibodies as kind of looking as like a Y molecule, kind of looking at a Y. And if we look at the arms of the Y, this is referred to as the FAB, or the antibody portion of the molecule. This is the actual portion that's going to bind to MHC or bind to antigen, this Y portion. This Y portion is connected to a stalk region that's referred to the FC, that's referred to as the FC region. And the FC region is actually going to be the part of the antibody molecule that's going to bind to cellular receptors and to help bind to complement proteins. We'll talk about complement in another lecture. So the antibody is basically two parts, the FAB region, which binds to antigen, and the FC portion, which basically binds to cellular receptors and kind of gives the antibody its biological activity, if you will. So we have 5 classes of antibodies within the body. They all share a basic common structure. And this common structure is useful because this allows antibodies to change types. So to go through, by going through a phenomenon referred to as class switching. So all antibodies carry a common structure. And within the body, we have 5 classes of antibodies that are referred to as Ig molecules, or immunoglobulin molecules. Immunoglobulin is the abbreviation for Ig. We have IgG, IgA, IgM, IgE, and IgE. And each one of these different types of antibody molecules is going to have a specific role that it performs within the context of antibody-mediated protection. IgG is the major antibody component within the body. So if you were to grab a handful of antibody from somebody and look at it, most of it's going to be IgG. It provides the majority of responses. For example, it's involved in placental passive immunity. So when we look at the immune response that the mother provides, protection the mother provides to the baby growing inside of it, a lot of that's going to be IgG. IgA is an antibody that's found primarily in mucosal areas, so areas around the ducts of the tears, the nares, the respiratory passages, and it's usually going to be found complexed in saliva or tears. Secretory IgA is a molecule, is an antibody molecule that's found a lot in mucosal areas in particular, and it's used to help provide protection against pathogens that use the respiratory tract to establish infection, such as influenza virus or certain types of respiratory bacteria. IgM is the largest of all of the antibody molecules. From a size perspective, it's very, very large. And IgM is expressed on the surface of all B cells. It's bound to the surface of B cells. And it's one of the first antibody responders before IgG responses can really become active. So IgG and IgM are going to be the 2 highest component antibodies from a molecule perspective found throughout the body. IgE is used to bind to allergens and trigger the release of histamines. Oftentimes when you're having an allergic response or someone comes in and they're saying, man, my allergies are really bad today, this is usually going to be driven through an IgE response. IgE is also very useful in providing protection against parasitic worms. We'll talk about here in a slide or two. And then IgD is an antigen receptor and a basic class of antibody that we see on unstimulated or unactivated B cells. So 5 classes of antibody, each of them provide a specific benefit, but they can also kind of crosswalk and help out and participate in other antibody activities within the body. And then, as I said it before, antibodies do have the ability to switch classes under certain circumstances. So an antibody that maybe started out as IgG can switch to become IgE or IgM through a mechanism called class switching, which we'll talk about in a future discussion. So when we look at the roles of antibodies within the immune system, they're really going to go out there and provide a couple functions. And we can break these functions down into neutralization, opsonization, or the activation of complement. So if we look at the cartoon up here on the left-hand side, we have our yellow antibody molecules, again, kind of designated with this cartoon upside down Y shape, and they're going to complex with bacterial toxins. So you have the production of antibodies, which can come in, and under normal conditions, these bacterial toxins are produced, and they're able to go out there and interact with cell receptors and then cause toxic effect to cells. But if antibodies are being produced, the antibodies can come along and they can actually bind up these little toxin molecules. And so the result is then the neutralization of the toxin, because now the toxins don't have the ability to physically go and bind to the receptor on the cell because they're covered with antibody and they don't fit anymore. And once the bacterial toxins are covered with antibody, they can then be ingested by a macrophage, and then broken down and removed from the host. We can also have antibodies being produced in the extracellular space that can go and coat bacteria. So in the middle panel, we see our blue antibody being produced. We've got some large rod-shaped bacteria that are in the extracellular space. And these rod-shaped bacteria are now going to get coated with antibodies through a process called opsonization. And now you have massive amounts of antibody all covering the surface of this bacteria. Now that the bacteria has been covered with antibody, it can be ingested by a macrophage and then broken down and removed from the host. And then the third role we see antibodies playing are, we have antibodies that are going to participate in the activation of the complement cascade. So we have our green antibody cartoon on the far right panel. We have bacteria that are maybe floating around in plasma. The antibodies are going to come along and recognize the bacteria in the plasma. They're going to begin to form complement. The complement system, which we'll talk about in a future talk, but for the purpose of this discussion here, complement is a series of proteins that are already protein factors that are present within the body that basically can be assembled into the ultimate production of a structure that will allow for the loss of membrane integrity on the surface of the bacteria. And once we have complement activation, eventually it's going to lead to the breakdown and destruction of the bacterial component. So we have antibodies that can come in and bind up toxins through neutralization and neutralize the toxins and activity. The antibodies can also come in very non-specifically and coat the surface of bacteria. This process of opsonization, which will then lead to the digestion and the intake and breakdown of bacteria by macrophages, and that we can have antibodies come in and activate complement, and then the production of complement, activation of complement leads to the production of structures on the surface of the bacteria, which will lead to the ultimate destruction of the bacteria. So antibodies participate in neutralization, opsonization, and the activation of complement to help drive an adaptive immune response. Antibodies are produced by B cells, the other primary cell component of the adaptive immune response. So we talked about T cells and the role that they play in helping to provide cellular immunity at the cellular level by eliminating virally infected cells. Now we have B cells that can come in along with T cells and produce antibody. And again, the role of antibody is going to be primarily neutralization, opsonization, and the activation of complement. And just as we saw with T cells, the activation of B cells to produce antibody is also going to require a co-stimulatory molecule that's absolutely critical in driving activation of the B cells. If we have B cells that are receiving the initial signal, just as we did with T cells to activate, but the B cell does not receive that co-stimulatory signal, then you're not going to have full activation of the B cell. And the B cell, just as we saw with the T cell, becomes anergic and basically inert, and it just kind of floats around. So again, this is another very elegant design to the immune system where we see an affirmative signal, an affirmative answer needing a co-signal to really confirm the response and activate. Just as we spoke about with T cells having the ability to generate a vast repertoire of T cell receptors to be able to respond to an almost infinite amount of pathogens, B cells carry a very similar mechanism that they rely on to generate B cell and antibody diversity. And this again, just as we saw with T cells and this VDJ, this recombination of these variable segments within the receptor, B cells use a very similar mechanism. So B cells have what are known as the B cell receptor, the BCR, and this B cell receptor goes through various rearrangements, and these rearrangements are extremely susceptible to mutation compared to regular DNA mutations. So I put this up here in the graph. Because of the ability of DNA polymerase to proofread, we generally see DNA mutations occurring that are about 1 in every million. So out of every 1 million bases that are replicated during DNA replication, there might be one mistake. but DNA polymerase has the ability to go back in and correct those mistakes. When we look at B cells and T cells, however, these processes are very susceptible to mutation, which when you think about needing to come up with a constant new receptor, it might be helpful to have the ability to go through more mutations than one would normally expect in DNA replication. And we see for B cells and T cells going through these receptor rearrangements that mutational rate goes from 1 in a million down to about 1 in 600. So basically, you're going to see 2 generations of mutations in every B cell population that leads to an expansion. So this again just helps to drive receptor diversity for B cells, which then is going to drive the various combinations you can get in antibodies. And again, both of these things are working in conjunction with T cells to kind of provide an almost unlimited amount of diversity within the immune system to respond against pathogens. So if you're on Mars and your T cells were having a hard time recognizing that new Martian bacteria, your B cells are going to have 10 to the 16 opportunities to go through B cell receptor rearrangements to produce an antibody that can recognize that antigen from Mars and help clear it from the host. So just as we saw with T cell receptors going through this VD-J recombination, we also have a process in B cells that we refer to as somatic hypermutation, which helps drive changes in the receptor and helps keep receptor diversity within B cells very high. Again, this gives the B cells the opportunity to be able to react and recognize a wide array of antigen, which is going to help provide a wide range of protection from the immune system protected within an infected host. So if we summarize the last 4 slides, just as we talked about with T cells, B cells are also capable of producing a diverse range of antibodies, which can bind to either toxin or toxin-producing pathogens, leading to the removal and clearance of those things from the host. So to kind of conclude this discussion on the immune system, the evolution of the immune system is probably exceeded by only the evolution of DNA as storage molecule for genetic information. If we don't have an immune system, we don't have a functioning host in a living organism. And the fact that the immune system, in one way or another, has been conserved across virtually every form of multicellular life on our planet shows that it is a vital component for multicellular organisms. If you don't have an intact immune system, you're going to have a really hard time functioning. When we look at mutations in the immune system within neonatal mice and even within humans that are prenatal, we often see that mutations in the immune system are lethal mutations. So if you have someone who's going to be, or an organism that's going to be born, and we've done these knockout studies in mice where we've been able to basically eliminate T cells. So those mice that are born, those transgenic mice that are born don't have T cells for the most part. They don't live very long. If we eliminate B cells, those animals don't live very long. We've done knockout studies with various components of the immune system, and we've showed pretty conclusively that you've got to have an intact immune system in order to have a functional organism. So it makes sense why the immune system has been so tightly conserved amongst all the multicellular forms of life on our planet, because we need an intact immune system to be able to function and survive. As the immune system has evolved, not only within our species, but within other multicellular forms of life on this planet, pathogens have evolved as well. I've often heard it described as biological warfare. So you have the immune system developing a strategy to deal with a pathogen, and then that pathogen develops a way to circumvent that strategy. And then the immune system comes and develops another strategy to counter that circumvention, and the pathogen finds another way around. So you have a constant system of evolution, examination, re-evolution, examination, re-evolution, where you have the pathogen and the immune system evolving together in many respects in order to try to leverage an advantage against the other. So we're very lucky, very fortunate that we've been gifted with a dynamic, elegant means to not only generate an immune response to preserve our health against virtually any pathogen that we see, but we also have the ability to save people around us from infection from the same pathogen by having an immune response that's able to rapidly clear and eliminate infections. And as we get further down in our discussions in the short course seminar series, we'll talk about some of the mechanisms that pathogens have evolved to evade the immune system, and what are some of the immune system's responses to deal with these attempts at immunoevasion. This concludes the first session for the short course for the Center for Genomic Research.